Swayam Prabha. Digital India. Educated India.
So any sort of change in terms of number of individual chromosome is called as aneuploidy. This change can be addition of chromosome or the deletion of chromosome, but one or two chromosomes, not the complete set of chromosomes. There can be various reasons for aneuploidy. For example, loss of chromosomes in the course of cell division due to deletion of centromere. Because of centromere, there is no spindle formation, spindle getting attached to the centromere and getting the two set of chromosomes separated and to form the new two progenies. There can also be loss of small chromosomes generated due to Robertsonian cross-location, which we just see. Then there is non-disjunction phenomena, the phenomena of failure of homologous chromosomes or sister chromatids to separate during the process of cell division by meiosis or mitosis. So there can be different reasons for this generation of abnormal number of chromosomes. There are different types of aneuploidy. For example, it is termed as nullisomy when there is a loss of both members of a homologous pair of chromosomes. For example, let us assume that there is chromosome number 11 which is getting lost. So both chromosome number 11 from the paternal and the maternal side, they get lost. This is being represented as 2n minus 2. So 2n is the diploid number, minus 2 represents that both the members of homologous chromosomes have been lost. So if you are talking about human karyotype, in total there are 46 chromosomes, minus 2 leading to 44 chromosomes in total in case of a nullisomic individual. It can be monosomic condition where there is a loss of single chromosome. So it is represented as 2n minus 1. Means in case of humans it will be 45 chromosome individual if it is monosomic. In case of trisomy, there is a gain of single chromosome and thus it is being represented as 2n plus 1, and hence in case of human it will be having 47 chromosomes if it is a trisomic individual. Same if it is tetrasomy, it will be gain of 2 homologous chromosome represented by 2n plus 2 and hence the 48 chromosomes. As you can see, there is a contrasting pairs nullisomy versus tetrasomy or monosomy versus trisomy. There can also be other types where more than one aneuploid mutation occur in the same individual. So here there can be example of double trisomy. That means there is an extra copy of two different non-homologous chromosome and thus it will be represented as 2n plus 1 plus 1. Double monosomic, there is a two fewer non-homologous chromosomes and hence represented as 2n minus 1 minus 1. Same is the case with double tetrasomic, which is represented as 2n plus 2 plus 2. That means two extra pairs of homologous chromosomes are there. So here instead of single aneuploid mutation, two or more than two aneuploid mutations are taking place but in the same individual. Now depending on whether this aneuploidy is with respect to the sex chromosomes or autosomes, there can be sex chromosomal aneuploids or the autosomal aneuploids. The famous examples you must have read in your books is Turner's syndrome, which is represented by X0 or XO condition, and the Klinefelter syndrome, which is XXY. So there is a Klinefelter male, there is a Turner's female. Both are sex chromosome aneuploids. Whereas autosomal aneuploids are there. The famous examples include Down syndrome, which is trisomy 21, Edwards syndrome, which is trisomy 18, Patayu syndrome, which is trisomy 13, and so on. Moving ahead, there is a condition called polyploidy. Now, this is also a numerical aberration of chromosomes, but there is a numerical aberration with respect to the set of chromosomes. Here, there is a presence of more than two genomic sets of chromosomes in contrast to usual diploid or two sets in most of the eukaryotes. So, if instead of diploid condition, there is an extra set of chromosomes, that means it is triploid condition, or two more sets, that is tetraploid, five sets in total, pentaploid, and so on. There can be autopolyploidy or allopolyploidy. Autopolyploidy means all the chromosomes set from a single species. This is due to the accidents which are occurring during the cell divisions, producing extra set of chromosomes. Whereas there is another condition which is called as allopolyploidy. Here the chromosomal sets are from two or more species. So there is a allo means from two different sources. Auto means from the same. In case of allopolyploidy, there is a chance that hybridization has taken place between the species, and that is why the chromosome sets from two different species are coming in the offsprings. There is an evolutionary significance of polyploidy with respect to overall size of cell and organisms. So if there is an increase in terms of chromosomes or chromosomal sets, it is going to increase the size of the cell or the organism as such. There are reports of genomic stability which is being acquired with respect to polyploidy. Tissue or organ specific gene expression, more genetic recombination, and therefore more genetic variations. So that is why we are considering polyploidy or chromosomal aberration here, because it is going to enhance the genetic variations. Now this is one example of numerical aberration in terms of polyploidy as a consequence of hybridization in the evolution of Nicotiana. Nicotiana tabacum, which is a fertile organism, it has the chromosome number as 48. So it is being represented here as 2T. When it gets hybridized with Nicotiana glutinosa, which is also fertile but having 24 chromosomes and representing as 2G, there is a sterile haploid that is being formed, which with the chromosome number 36. So from this diploid 2T, haploid set is coming here. So that means 24 chromosome from here. This diploid is giving haploid set in terms of 12 chromosome here. In total, it makes 36 chromosomes. There is a chromosomal doubling here, which gives rise to Nicotiana digluta, which is a fertile organism, but the chromosome number has increased to 72. So this is 2T plus 2G. So instead of 48 chromosome here or 24 chromosomes or 36 chromosome, now this plant has 72 chromosome, which is a polyploidy situation. But this shows how this plant evolved because of hybridization and polyploidy. Now, after the chromosomal aberrations, let us move ahead to the gene mutations or the point mutations and how these point mutation or gene mutations act as a source of variations. There can be various types of mutations or gene mutations which we are going to discuss in the next few minutes. The definition, if you talk about gene or point mutation, it can be given as any sort of change in the structure of gene which is often related to the function of the genes as well, or in other words, change in the nucleotide sequence of the genetic material is referred to gene mutation or the point mutation. Let's talk about the types of these mutations. There is first category which is called as base substitution, where one base is getting substituted by another base. there are two different categories or two different possibilities which can occur in the base substitution, which includes the transition and the transversion. Now let us say if this is the starting sequence, this is a DNA structure if you can here, based on whether a purine is being replaced by another purine or pyrimidine is being replaced by another pyrimidine or vice versa, there can be transition or transversion. In case of transition, a purine is replaced by purine and pyrimidine is replaced by pyrimidine. Whereas in case of transversion, a purine is replaced by pyrimidine and a pyrimidine is being replaced by purine. So this is the major difference between the transition or transversion, though both are base substitution. Let us see in more elaborate situation. Here you have both the purines, that is adenine and guanine, and here at the lower bottom side you can see the cytosine or thymine, which are the pyrimidine. So if it is adenine to guanine transition or substitution, this is called as transition type of base substitution. If you have cytosine to thymine kind of change or substitution, it is also called as transition. Here it is purine to purine and here it is pyrimidine to pyrimidine. Whereas any other change, that is from A to T or from G to C or A to C or G to T and vice versa, where there is a change or substitution between purine and pyrimidine or vice versa, it is called as transversion. So with this simple representation, you can try understand the difference between transition and transversion. Any purine to purine or pyrimidine to pyrimidine change is transition. Any change between purine to pyrimidine or pyrimidine to purine is called as transversion. Two more kind of point mutations or gene mutations which do exist are called as insertion or deletion. As their name suggests, in insertion we do have an addition of nucleotides, whereas in case of deletion we have elimination of some nucleotides from this starting sequence. The gene mutations or the point mutations can be classified as various types depending on the kind of change that is taking place. Let us try to understand the three basic type of mutations which are called as silent mutations, nonsense mutations or the missense mutations. Let us try to understand them at all these three levels, that is the DNA, mRNA and the protein. If there is no mutation, this TTC codon gives rise to AAG and it ultimately forms the lysine as the amino acid. Now let us see different kind of mutation. If a mutation occurs which changes this C, which is at the third position of this codon, to T, that means the codon has changed, mutation has taken place, and if you see at the protein level, same amino acid is being formed because the change is at the mobile position and there is always a degeneracy in our genetic code, because of which both of these codons are forming the same amino acid. So there is no change in the amino acid, means the mutation that has occurred, it is having a silent kind of effect. So they are termed as the silent type of point mutations because the same type of amino acid is being formed, and we know the reason behind this is the degeneracy of the genetic code. There can be another type of mutations which we call as nonsense mutations. Here in this TTC there is a mutation which converts this first T with the A, because of which there is a UAG codon which is being formed, which is nothing but a stop codon. That means it terminates the synthesis of the protein. So there is no amino acid is being formed from here, and that is why it is called as nonsense kind of point mutations. Then there are mutations which are called as missense mutations. That means they change the sense of amino acids or the proteins as such. The missense mutation can be of two types. One is called as conservative, another one is called as non-conservative. In conservative, there is a change or mutation, and the type of amino acid which is being formed, it is having almost similar properties in terms of physicochemical properties of the initial amino acids without the mutation. So this is called as conservative type of, though it is missense mutation, it is changing the amino acid, but the amino acid which is being formed now, it is having the similar kind of physicochemical properties which were there in the amino acids which is to be formed without the mutation. The non-conservative missense mutation forms an amino acid which is totally different from the original amino acid. So, depending on what kind of amino acid is being formed, they can be conservative or non-conservative type of missense mutations. Then there is another classification which is being narrated in various books, which is called as silent versus neutral mutations. Again, if this is the situation without any mutation, we just discussed about the silent mutation. That means instead of lysine, again there is a new amino acid which is again the same, lysine only. Hence the effect is silent and it is called as silent mutations. Where in case of neutral mutations, there is a different amino acid, but this amino acid is having the similar properties as the one which was there without mutation, as both lysine and arginine are the positively charged amino acids, basic amino acids. So this is called as neutral mutations. These neutral mutations are nothing but the conservative type of missense mutations which we just discussed in the previous slides. Mutations can also be defined as loss of function mutation or gain of function mutation. The loss of function mutations cause the complete or partial absence of the normal function. So if a mutation occurs because of which there is a loss, complete or partial, of the functioning of the amino acids or the protein, this is called as loss of function mutations. They are frequently recessive, and diploid individuals must be homozygous for the mutation before they can exhibit the effects of the loss of the functional protein. Because otherwise, if they are heterozygous, the normal copy of the protein will dominate here and you won't be able to see the effect of loss of function mutation. In contrast, there is gain of function mutations which produce an entirely new trait or causes a trait to appear in inappropriate tissues or at inappropriate times in development. This is called as gain of function mutation. So there is a new acquisition of a new trait. They are frequently dominant in their expression. So even if the individual is not homozygous for it, since they are dominant in expression, you will be able to see their effects. The mutations can also be either the conditional mutations, that means they are expressed only under certain conditions. So there are certain conditions under which you can see their effects. Or they can be lethal mutations also, which cause premature death also. These are another type of classification which is being used for the point mutations. Also, the point mutations or gene mutations can be classified as forward mutations or the reverse mutations. The forward mutation changes the wild type phenotype to a mutant phenotype. So if it is a wild type phenotype and a new mutation is occurring which is causing it to be a mutant type, then this is called as forward mutation. Whereas in case of reverse mutation, the mutant phenotype changes back to the wild type phenotype. So here you can see if A is the wild type here and A plus denotes the mutant type, in forward mutation this wild type gets converted to the mutant 1, whereas in case of reverse mutation, this mutant A plus gets back to its wild form, that is A. Now, this gene or point mutations can also be the suppressor mutations. Unlike the forward and reverse mutation case which we saw in the previous slide, in case of suppressor mutation, there is a suppression of effect of an earlier mutation but at a different site. That means there is a second mutation which is not the reverse mutation, but it is occurring at a different site, but it suppresses the effect of the previous or earlier mutation, and hence they are called as suppressor mutation. So the major difference between the reverse mutation and the suppressor mutation is, in case of a reverse mutation, there is a reversal of the effect, but the mutation is occurring at the same site. Whereas in the suppressor mutation, there is a suppression of effect of previous mutation, but they are occurring at a different site. The suppressor mutation can also be classified or differentiated as intragenic suppressor mutation or intergenic suppressor mutation. In case of intragenic suppressor mutation, there is a second mutation within the same gene which restores function of the mutant gene product. Whereas in case of intergenic suppressor mutation, there is a second mutation which relieves the effects of a mutation in one gene by mutation somewhere else within the genome. So here it is the same genome, the same chromosome, but here it is the some other location within the genome. Here it occurs in the same gene where the primary mutation exists. The location might be different but within the same gene. Whereas in case of intergenic suppressor mutation, it occurs somewhere else in the genome other than the gene within the primary mutation. The intragenic suppressor mutation relieves the effect of primary mutation within the same gene, whereas the intergenic suppressor mutation relieves the effect of primary mutation somewhere else in the genome. They provide information about the structure-function relationship of the proteins, whereas intergenic suppressor mutation provides information about functionally related proteins to the primary mutant. So these are the basic differences between the intragenic and intergenic suppressor mutations. Then there is another category which is called as spontaneous mutation or the induced mutation. Spontaneous mutations are the result of natural internal changes of the DNA structures. This can be the spontaneous replication errors, the tautomerism, depurination or deamination phenomena. Here there is no external source of causing the mutations. Whereas in case of induced mutations, there is a change which is associated or which is being caused by the external factors, and these external factors are most of the time environmental chemicals or the radiations. So there can be a chemical source or chemical mutagens, or there can be a physical mutagens or physical source of mutations. Under chemical mutagens, they can be a base analogs, that means the base like chemicals which substitutes the bases. They can be base modifying agents which changes the bases, or there can be intercalating agents which get fixed in the DNA or they get intercalated within the DNA. So any of these cause the abnormality with the normal base pairing, and hence they cause the mutation. Whereas there can be physical source of mutation as well through the most common example is the UV radiations. Now let us quickly summarize what we discussed in this lecture. We talked about genetic variations and their role in evolution. Then we talked about various types of genetic variations based on the categories, that is cells affected, the degree, the impact, direction of evolution, and so on. And then we started discussing about the sources of variations, that means at individual level or population level. So far we have covered the individual level of sources of variation, that is the chromosomal aberrations both in structure and the numerical or the number of chromosomes or the set of chromosomes. And we have also discussed about the point or gene mutations. In the next lecture, which is second part of this, we are going to discuss the remaining individual variations, and we are going to discuss the source of variations at the population level. It means the sources which affect the gene pool of a population. We are going to discuss all these sources. Also, we will discuss the points of importance of variation in the evolution. You can refer these books on evolution, and also you can refer various books on genetics, particularly the book called Genetics, a Conceptual Approach by Benjamin E. Pierce for all these chromosomal and gene mutations. Thank you so much.